Assalamu alaikum. Today we are going to study some main topics of subject biology class first year. These video lectures are also useful for MBBS test. These lectures are in very simple words by which anyone can take help. If I miss anything in this video or you don't understand anything, please comment below. So let's move to our lecture. Let's explore some questions of biology with answers. What are enzymes? Enzymes are biological molecules, typically proteins, that act as catalysts in living organisms. Catalysts are substances that increase the rate of chemical reactions without being consumed or permanently altered in the process. Enzymes play a crucial role in metabolic pathways and various cellular processes by facilitating specific chemical reactions.
What is the structure of enzyme? Enzymes are typically proteins, although some RNA molecules can also exhibit enzymatic activity. They have a complex three-dimensional structure consisting of one or more polypeptide chains folded into a specific shape, often with a crevice or active site where the substrate binds.
How does a enzyme catalyze a reaction? Enzymes lower the activation energy for a chemical reaction to occur, thereby increasing the rate of reaction. They do this by binding to the substrate, facilitating the formation of the transition state, and providing an alternative reaction pathway with a lower energy barrier. What are the factors that can affect enzyme activity? Several factors can influence enzyme activity, including temperature, pH, substrate concentration, enzyme concentration, and the presence of inhibitors or activators. Changes in these factors can alter the enzyme's structure or the kinetics of the reaction, thus affecting its activity. What is enzyme inhibition? Enzyme inhibition refers to the process by which a molecule called an inhibitor binds to an enzyme and reduces its activity. Inhibitors can be reversible or irreversible, and they can bind to the active site or other allosteric sites on the enzyme, disrupting its function. How does competitive inhibition works? Competitive inhibition occurs when an inhibitor molecule competes with the substrate for binding to the active site of enzyme. Inhibitor and substrate are structurally similar, and by occupying the active site, inhibitor prevents the substrate from binding, reducing enzyme activity. What is non-competitive inhibition? Non-competitive inhibition involves the binding of an inhibitor to a site other than the active site, called an allosteric site on the enzyme. This binding induces a conformational change in the enzyme that reduces its catalytic activity, regardless of substrate concentration. How are enzymes classified? Enzymes can be classified into several groups based on their catalytic activities and the reactions they catalyze. Common enzyme classes include oxidoreductases, transferases, hydrolases, lyses, isomerases, and ligases based on the type of reaction they facilitate.
What is feedback inhibition? Feedback inhibition, also known as end product inhibition, is a regulatory mechanism in which the final product of a metabolic pathway inhibits or reduces the activity of an enzyme earlier in the pathway. This mechanism helps maintain homeostasis and control the rate of overall metabolic pathway.
What are oxidoreductase? Oxidoreductase are enzymes that catalyze oxidation reduction reactions involving the transfer of electrons between molecules. Examples include dehydrogenases, which remove hydrogen atoms from substrates, and oxidases, which transfer electrons to oxygen. What are transferases? Transferases are enzymes that facilitate the transfer of functional groups such as methyl, phosphate, or amino groups from one molecule to another. Examples include kinases, which transfer phosphate groups, and transaminases, which transfer amino groups. How does enzyme classification help in understanding enzyme function? Enzyme classification provides systematic way of organizing and categorizing enzymes based on their specific catalytic activities. Understanding the enzyme's class and its associated reaction can give insights into its mechanism of action, substrate specificity, and biological functions. What is lock and key model? The lock and key model is a concept used to explain specificity of enzyme substrate interactions. According to this model, active site of an enzyme is like a lock and the substrate is the corresponding key that fits into the lock. The active site has a specific shape and chemical properties that perfectly complement those of the substrate. What is induced fit model? The induced fit model is an alternative concept that describes the interaction between enzymes and substrates. It suggests that active site of an enzyme is not completely rigid but rather undergoes conformational changes upon substrate binding.
What is effect of pH? pH plays a crucial role in influencing the structure, activity and stability of biomolecules including enzymes. The effect of pH on biological systems can be summarized as follows. Protein structure and function. Proteins including enzymes have an optimal pH at which they exhibit maximum stability and activity. The ionization state of amino acid residues in the protein is influenced by pH, which in turn affects the protein's structure and function.
If you like our video, then please like, subscribe, comment and share our channel Dr. Raza Academy and Educational Consultancy.